really not enough people to do this, I want to talk about graph 5. So how to do graph 5, because it's the only graph that we have not done yet. But maybe I'll just bring it up and then we can think about it later. Okay, so this is what we were going to graph for graph 5. Let me bring it up here. Okay, so here's.
Sorry.
Oh, that's okay. So here's the data table that you have already filled out. So you have your optical densities for whatever your cultures were. And then you also have your, let's see, I think this is the one that we filled out. You have your dilutions, but I think there's another place where you fill out what your dilutions are. Down here, sorry. So you'll have your optical density readings, and then you'll have your dilutions. So the reason that we did this is we actually took a culture, and we didn't know how many organisms was it, so we actually used the spec to figure out the optical density. Okay, so we have the OD over here, and then because this will be this optical density, and say this is pretty dense, maybe this 1.05 times 10 to the 6th or whatever it is, and then this would be a little less, maybe 0.85 or 80 times 10 to the 5th, and this could be 0.750 times 10 to the 4th. And anyway, we'll get down to maybe a low density. I don't know how it ever got. I think all of ours were pretty high, but this is normally how your OD would be. It would be get less and less and less. And then down here, where it has 1 to the 16, 1 over 8, 1 over 4, 1 over 2, and 1 over 1. This would be our colony forming units per mil calculations that you did. So over in the second column is where you had your colony forming units that you determined, and you only need one countable plate. And that countable plate would have told you how many colony forming units you have per mil. Okay? And so now that you know that, that's what you have in all the mils you had. This is all in one mil that we took our spec on. So for this one is your undiluted, so that would be 100% of your first calculation that was colony forming units per mil. And it was probably 1 times 8 to the 10th or something. I don't know how many colonies that you got. You would have some large number of colonies. And then, to be able to put it on this graph to compare to this, you would dilute it by 2. So you would take this number and divide it by 2. You divide it by 2 again, divide it by 2 again, divide it by 2 again. That's exactly what we did. Undiluted, divided by 2 is a 4, divided by 2 again is 8, divided by 2 is 16. So this is a way that you can go along the bottom of this graph and you can put your calculated values of the colony forming units. So see, you have 1 to 16, 1 to 8, 1 to 4, 1 to 2, 1 to 1. So that way you can put what the colony forming units are, and then from your graph would be something like maybe this and that and that and that and that and that, and you would have some line best fit that you would say, okay, if I have an OD reading at this, I'll go to the line, I'll go down here, and I'll figure out, because these will all be in colony forming units, I'll figure out what my colony forming units are. So that is the reason that we did our analysis. And we used an E. coli. I think that's what we used. And the E. coli, we used it for this, and then we used it for this. So you have to use the same organism grown at the same OD so that you can do this graph and then do this graph where you actually do your dilution. So this tube that we had for the E. coli was both diluted to do for the spec, and then also we made dilutions for the colony-forming units per mil. Okay, so you should give that a try, and I'll set a time when everybody feels comfortable, they can turn it in. Not many people are here, so we'll have to repeat it to them. But maybe on Thursday, we'll talk about it again, and then we'll set this for next week. And that will get this lab 5 out of the way, because that's our only outstanding one that we don't have done. Okay? Okay, and does this somewhat make sense what we're doing? This is the OD that we took. We're trying to figure out what these OD is for this particular organism at this particular time. And so we actually took it and we diluted it and made plates, and we could count on the plates. And then we figured out our colony-forming units, and then we can match on our graph. It's not a really useful tool, because how often are you going to use the exact same one and then plate it and then figure out? Because, you know, if you go to use another culture, you have to do the whole thing again. So you have to have the culture, the two cultures match. So it's not like you can say, okay, for E. coli, I'll always use this particular graph, because it won't be for whatever E. coli you have. So it's just a method for us to say we needed to figure it out. We could figure out how many colony-forming units there were at any point on the graph. Yes?
Should our graph be linear when we're doing this? Or
Well,
should it be exponential?
In theory, it should be. But what happens with this graph is when we took our spec were really high, most of them. You didn't have a nice gradation. You didn't have to go from 1 to 8 to 7 to 6 to 3 to 2 to 1. You went from 1 something to 1.04 and 1.8. I mean, just all of it was all grouped up here. So you didn't really get a very good graph. And that's because we grow stuff for 48 hours. So the bacteria, they multiply every 20 minutes. So the bacteria are just way overgrown. So we didn't really get a good graph. The ones that we did on the plates was fine, but we don't really have anything to kind of graph it to. So you'll graph it just to turn it in to say, I understand this could be a good way to do it. Let's say if you stop the growth, maybe the tech came in at 19 hours and stopped the growth, and then we use that 19-hour culture. That's usually what they do. It's usually 18 hours, 12 to 18 hours they use for the culture. But this is, we grew them for a couple days. Yes?
So to create this graph, we don't need to watch the video. The video is for Lab 13's graph? Or for both graphs?
No, the one for 13 is not this type of graph at all. This is a hand-done graph. This is, let me show you on here. This is this graph that you can see right here. And this is purely hand-done. Yeah, and so it even gives you the little marks. This is where you're going to do it, put your colony-forming units, your dilutions, and then this is your optical density up here. So the only thing you have to do by hand is try to figure out how many little square things can get whatever ODs you have in some kind of order. And you don't want to, once you set your scale, you don't want to alter it to say, oh, I want to get another one in. You have to have the same ratio from this one to this one to this one to this one. Otherwise, your graph isn't useful.
So then Lab 13 we'll do on the computer, and then we'll copy it to this sheet.
Lab 13?
Lab report 13.
That's separate. These are completely different.
I know this is Lab 5, but
yes.
for Lab 13, the one that we have to watch the video.
Oh, that is just for helping you to graph, and that is something that it just gives you data to show you how to graph.
Okay.
Yeah, that's purely it. It really doesn't have much to do with anything in the lab that we did because our growth was so overgrown.
Okay.
Yeah, so you don't, you graph that to learn how to do the graphing.
Okay.
And then later on, at least last semester, I was given a notice, and I don't know what department gives it to us, for learning objectives that students have in classes. So they said, you are scheduled to do a learning assessment. I go, okay, what is that? And they said something that was on the learning assessment list. You need to give it to your students unannounced, and they need to be able to do the learning assessment, and they turn it into the learning assessment place here, and then they assess whether students are learning what they should be learning, how they should be learning it. So I go, okay, so I had to call the coordinator, and I said, what in the heck is a learning assessment, and what did we do that's a learning assessment? And he goes, oh, this is an easy one, have your students do that. And I go, oh, okay, that'll be good, because we haven't done it. And so on one of the quizzes, I just said, okay, take out the computer, here's some numbers, you put it in, you make a graph, you email it to me, and then that's what I turned in. Yeah. So, I don't know, I hear that the learning assessment is given to different professors at different times, so maybe we won't have it this year. But I do want to make sure that we do this in case this is something that we have to do. Okay, yes?
Instructor, so just to clarify, so for this graph here, we'll be using this information here, right? To plug that in?
I'm not sure what
This is the page you had us fill in with when we did our count.
Yes.
Right?
So this page that you filled out, in theory, if this were a perfect world, these would be 10 apart, right? So this, you had tenfold dilutions, but some of them are not that. You get something that's 2.6 times 10 to the 8th, 2.9 times 10 to the 7th, that's pretty good, and 6, 5, okay, so these look pretty good. Sometimes you see them and they're way off. It's just because our pipetting, we're not very good with that. So this part here, you pick the best one.
Okay.
So they're all, because they're tenfold dilutions, you want to pick the one that is 1-to-1. So this one down here, so this is probably what the original culture was. Okay, so this one, so 2.68 times 10 to the 8th. So that's what you would have in this square right here.
Yeah.
So that's a 1-to-1, that's what it is. And then you would dilute that by just dividing it, not any number-wise. You would dilute it by 2, and then it would be a 1-to-2. And then you dilute it again, and that's a 1-to-4. And you dilute it again by 2, and that's a 1-to-8. And you dilute it again by 2, and that's a 1-to-16. Okay. So each time you're doing that, you are going to get a number of colony-forming units per mil.
Okay.
Then that number is going to go down here on the graph where this is. So this bottom line is your dilutions, your colony-forming units per mil. So the 1-to-16 will have whatever you have in this square, your colony-forming units per mil. And your other ones, the 1-to-8, 1-to-4, 1-to-2, they would be all in this square.
Okay.
And these different squares. So then if your OD was really good, you would have some graph on your OD that because this is 1-to-16, your colony-forming units per mil would be relatively low. And so your graph would go somewhat like this. I mean, it wouldn't be a perfect graph, but that's what you would expect. But all the ones that I've seen, like last semester and something like that, they had a huge drop up here, and then it went up and down and up and down. But it's just something you just need to do.
Okay.
So the conceptual idea is that you can take a culture that you don't know what the colony-forming units are. You can take the OD, you can graph the OD, and then you can do dilutions, and you can take these dilutions with colony-forming units that you plate on a plate, and you physically count the colonies. You would be able to make some dilution to say, oh, okay, in this one, it was so many colony-forming units, and this one, it was, X, Y, Z. Okay, you would just go from the line, whatever line we had, let's say we've got something else over here, we go to the line and go down and figure out what the colony-forming units were. Or if you had something over here, you go down and figure out the colony-forming units.
Okay.
Yeah, it's just a tool that you need to know that is possible to do.
Question.
Uh-huh.
So how do we determine which one to pick again?
You want to do the one that has the highest colony-forming units per mil.
Okay.
And that's usually your 1-to-1.
So since we were on the same team, bro, so one here in the corner with I believe we ended with the 2. I don't have my glasses, so I think it's 2.64.
Okay.
Yeah, it's usually your highest reading, and I mean your readings look really good. They were about tenfold different between each one, which we did tenfold dilution, so that seems like it should be somewhat right.
So that's what you put on the 1-to-1 undiluted, right here?
Yes, and then from that,
The 2.6 or the 0.0001.
so this
The same,
this
the same thing.
this form that you have, that's what you would put right here, the very top one. That's
So
the 1-to-1 undiluted.
And for optimal density reading, how do you get that through this?
Well, that's the density reading that you took. So these densities up here, you took with the spec, and you wrote them down.
I think,
Yeah.
is your question that, are we using the CFUs or are we using the final scoring, which is the 0.0001? Or the scientific would be 2.6 to the 10, or to the 8, sorry.
So optical density goes here, these goes from the tubes that we got from the spec, we have a spec reading, and so that's what goes here. And then down here is your dilution, and your dilution is from here, you divide it in 2, and then you divide that answer in 2 again, and you divide that answer in 2 again, and you divide that answer in 2 again.
So we're basically doing is the first undiluted number, we're just dividing that by 2, and then that.
And divide by 2 again,
That's
yes.
what-
Yeah.
And so really what we're doing, we're saying undiluted, and then we're saying divided by 2, and that's going to go right here, and divided by 2 again, and that's 1 to 4, and divided by 2 again is 1 to 8, and divided by 2 again is 1 to 16. So that's kind of where it's been mapped out to put on this graph. Okay.
Yeah.
And so everybody's set with that? So I will announce it again on Thursday, and I'll probably make it due the next Tuesday, and that way we just get this lab out of the way. Because it's just a matter of a little math, and you've got all the data. It's just been sitting there for a long time. A couple people have kind of tried it, and they brought me what they did, and it was not correct. So I just want to make sure somebody doesn't turn in something that's incorrect.
Thank you.
Okay, so you've got that? All right, so the other thing I wanted to mention, everybody is, for the graph on lab 13, we've got that all set. Okay, so we don't, I don't like to necessarily take lecture time, but it's really hard in the lab to get everybody so that they can listen at the same time. Okay, all right, then today what we're going to talk about is chapter 9 and chapter 7. There'll be an overview of chapter 9. It's a very dry chapter. It's talking about what kind of chemicals you use to kill bacteria. And so it's just chemical after chemical after chemical. So I'm going to give you a table that's going to be important for the quiz from my point of view, and then just a couple other things on that on chapter, so you'll have a good idea of what is important to review. And then chapter 7, that's what we're going to lecture on today. There's a lot of information, and I'm going to parse it down to something that's somewhat reasonable. There are a couple of really interesting things in there, and we can talk about those. And then study guides. I think somebody asked about study guides. So tomorrow is probably the first day I will be able to do them, and it will include the chapters that we go over tonight. So I think we have a Tuesday and a Wednesday or something quiz, or maybe it's a Thursday and a Monday, Thursday and a Wednesday. I'm not sure what the quizzes are.
I think it's Wednesday, Thursday.
Wednesday, Thursday, okay. So that should be, if I give it to you Thursday and it's Wednesday, I think that's 6 subjects. So that's about the best I can do. And then the microscope skills test, we will continue that. There's a list that I have of things that are checked in the microscope. Let me see, I went and got that out of the lab. So this is not something that we share, but I want to make sure that you understand this is a skills test to find something in the microscope, so that is the crucial part. But they have things like how you handle a slide, and could you focus it, and did you use the right paper to do it, and did it take you longer than 3 minutes to find your object, and was it very dim, and did you squint with one eye, and I mean, there's a whole list of all kinds of things that I'm checking when I'm watching you do this. And so far, they've been pretty good. So it's not, I thought when they first did it, I said, oh, I'm going to change mine to 5 minutes. But some people were up there like 2.5 minutes, and they were done. So I am going to say, for mine, I'm going to say 5 minutes is a max for somebody to find something. But really, 3 minutes is what our guideline is. That's what we should be taking. Yes?
What's a common mistake that students make during this microscope skills test?
They usually no, I don't want to tell you. I don't want to tell you, because then I'm giving people who have not done it yet, I'm giving them an advantage to say, then make sure they don't do that. Yeah. But everything that's on here is in that very first paper that you got. It says, how do you handle the microscope, what do you do, etc. So it's all written in there. And pretty much, I think everybody is handling the microscopes well. So, Yeah, the issue, it seemed like that we had, and it's not a microscope issue, it's not cleaning the slide well enough. So people were using the slide before you, and so now I got 2 slides so that one person that's done with the slide will come off and let the other person take the clean slide and start looking. And then they will clean that other slide really well, and so they'll put it there. So now the person that's done with this slide will clean theirs really well, and then this can go out. Because the very first person had no problem. And I think second, when somebody went, maybe the slide was not cleaned well enough, because it seemed like that was the issue on finding. Even though we got another slide, but it still was an issue. So now I have 2 slides that he has given me, and we'll just rotate slides so they can be cleaned really well. Everything else, we're going to use that microscope up in front, which is nice. Everybody's kind of zipped through it, so I think that's pretty good. Yeah, so anyway, this is the microscope thing, and we'll continue doing that. There's no deadline, although I'm sure everybody, because it's 30 points, so everybody wants to see some kind of point value, a high point value like that in your grades. So I would like to get it done on Thursday. Thursdays, there's quite a bit to do. It doesn't seem like in lab, but you're taking all the worksheets that you're having, you're filling those worksheets in with the data that you get from your results. So it does take a little bit of time. And then the next thing I'm going to do with that is, because this is part of a 25-point project, there's no lab report that you turn in. The lab report that you have is a report of everything that you did, little drawings and things, and then it's turned in as a project. So there's no data that I can put in, or score I can put in for lab report for you. So I'm going to have you turn in there's that worksheet that looks like, maybe I can find that on here. So for 14, so there's this sheet that's right here. Oh, where's this one? This one. So you have one of these, and I'm going to give you a brand new one, and each person will fill out just one column of this, so that you can tell me what your unknown number was. It's the same as your partner's, but you're going to put down what you did. What did you see in your microscope? Everything that you did, and then you're going to turn this in, and you're going to tell me what you think this organism is. And your partner will do the same thing. And yes, you're going to talk about it and stuff like that, but the worst thing to do is if you think it's one thing, and then your partner says, no, I think this test was like this, and you go, oh, okay, I'll just change mine to what you have, then you both might be wrong, or one of you has influenced the other one. So there'll be some points value of this, not very many, but at least some that we can put in. So anyway, I'll have you do that, but it's all with the data that you already have. Okay, and then I think the whole packet is due April 4th, or it's a couple weeks away, or maybe even a week away. I guess it can't be too much. Okay, makes sense? All right, and as far as I know, I talked to the tech, and everything is basically gone now. All of the reagents and stuff are gone. So it's, if you go back and your partner has one result for a test and you have something different, there's not going to be any way we can resolve it, because I think the materials are all gone now. So anyway, that's you have to almost find it at the spot, because this is only a 2-day lab. The one that we do for the gram-negative is something like an entire 2 weeks or something. And the beauty of that is maybe you have 15 or 18 tests that you do, and you start identifying your unknown, and one of the tests that you did does not match with what you think your unknown is. So what you can do is you can go and get another one of those tests from the tech, and you can run it again. And that way it will confirm to you, was your first test correct? or was the other test correct? And then if that is, maybe that's going to change what you think your unknown is. That one is, I'm going to think it's 100 points or 75. It's a large amount of points. This is 25. So it is a lot of points, but it's not huge. So by then, you're really pretty much an expert at all these tests that you'll have to do. Okay, so anyway, this thing I will then bring enough for everybody to have one, and you will turn it in on Thursday then after you've read all your results and you've made your decision. Okay, and then I'll grade that, and that will be considered a lab report, but probably a little bit more points than a regular lab report. Maybe it'll be 5 versus 2, because there's a lot of work involved in this. And then you'll do your group project together with your whole group. Okay, so that was that. Okay, I think that's all I wanted to make sure I told you. All right, so let's go ahead and maybe get to a little bit of the lectures. And so do we have all these people are gone? Okay. So anybody not pick up? Okay, so you want to come pick yours up? Yeah, so wow. So a couple people emailed me, but not all of these people. Did I take yours? Oh. Okay, well. All right, let's take the lectures. So I'm going to go to 9 first, and this will be where I'm really truncating a lot. I just want to point out things that you should focus on, which is number 1, this slide. So almost everything that follows this, is going to be individually discussing each one of these. So this is a good one to have to just kind of go through. Okay, so it kind of summarizes everything that we're going to do. And if you have any questions, you want to look at pasteurization or sanitation or something, the following slides will give you more detail. All right, so I will talk about a couple of these things, but this is, antiseptic sprays and stuff like that, and these are just kind of bullet points, and I could go through and I'm really just reading to you bullet points. So I think most of this stuff you can go through on your own, and then this is the big part that you want to focus on. This is really when you're plotting the death rate, and the big thing we have here, you can see that these are a magnitude of 10 apart. So if you're just looking between here and here, that's really a 10% of something. So here they're showing that you had 10% died between here and here. So and it took 1 minute for this to happen. And it seems a little awkward because it's only going between this and this. And you think, oh, 90% died? Well, this is the survival. So if this is 10%, then that means 90% died. Okay, so these are all intense, and this is all the living organisms, and this is the time frame. So you can see as the time goes down, more and more, 10% more, 10% more, 10% more are going to be dying off. So all this is just a graph just to kind of plot how organisms can survive. And here, we haven't talked, and it's still several chapters away, but it's starting to get important for us to understand that we can kill microorganisms for several reasons. And those reasons are things that the organism is vulnerable to. And one of the things we have is, if we have an organism like this, the first thing that we have is the cell wall. So let's say here's an organism, and if we have peptidoglycan layers like this, on it. What kind of organism is this? If that's the peptidoglycan layers and this is the organism. Well, let me give you another organism. I drew that one too big. Here's another organism that has, and then it has like this. So this is the membrane here, and here's the membrane, and then it has like this. So what would you call this one versus this one? And this is also its membrane right here.
The first one gram positive?
Yeah, so this is the peptidoglycan. Okay, and then this is the gram negative, and here's the peptidoglycan in here. So drugs and antibiotics, they target useful things inside the bacteria. So they have ribosomes like this they can target. They have DNA that they can target. They have different things inside the cell that can be targeted. So all of these types of things, like this is damage to the envelope interrupts viral replication. So whenever you damage things, part of the cell, that's going to cause the cell to start being compromised. And when we think about antibiotics, antibiotics are big molecules that are like this or something. In proportion, that's probably much too large. But here, if we're going through a cell wall, and let's say this antibiotic has to act on the cell wall, on the peptidoglycan, this is really much more vulnerable than this one is. Because this large molecule would have to go through this outer membrane to ever get to the peptidoglycan. So when we talk about what these antimicrobial agents are attacking, we have to kind of draw and think about the organism itself and then see which one would be more vulnerable. And of course, this chapter is talking about not only this, but they're talking about archaea, they're talking about viruses, they're talking about a lot of things. Okay, so this is just saying if you disrupt the cell wall, which really makes a lot of sense, then the cell is going to burst because this is protecting it from the osmotic pressure. Okay. And other things, the cytoplasmic membrane, and that would be this in here. This is the membrane that's right against the cytoplasm. When it's damaged, then all the contents go down. So it's good to me to maybe have a little drawing and kind of visualize what each of these statements mean. And these are viruses, and we haven't talked about them, and viruses are just non-enveloped. Some of them are enveloped, and some are non-enveloped. And we'll talk about them a lot more. So I'm not, this information are not, viruses you are not held responsible for until we actually talk about that. This is action on the proteins, and so we have nucleic acid, and then from the ribosome, this is where we make proteins. So mRNA comes through here, and then we start making proteins, and they fold up however they're going to fold. So targeting the ribosome is something that drugs can do that can knock out a bacteria, because if it can't make its proteins, it can't survive. Or it can actually target the protein itself. I could talk about maybe amino acids or something that they can alter. But this is really a lot of heat, and that would cause this to denature. So that's one way you can kill organisms. So there's lots of different ways that if you want to hinder the organism from growing, you can look at protein synthesis. You can look at maybe doing something that's going to cause mutations. I think we're in here. So you can cause mutations. We're going to talk about a little bit about that in 7. Also next, and I'll get to your question in a second. So next lab that we do after this one, we're going to do, I think it's UV radiation, and we're going to radiate organisms and we're going to see how it affects them. So it will be pretty surprising how some of them will be able to grow, and they have enzymes within them, some bacteria, that they can repair that damage if you put it in light. So they have light-sensitive enzymes that will take the light, and then they will use that energy to repair the damage that's just been done by the UV. If you don't put the organism in light, the organism will start to deteriorate because the UV destroyed them. So you can repair those mutations that are made by the UV if the organism gets back in light. It's really kind of amazing that they can do that. It's only certain organisms. Okay, so here, I think this is kind of logical, kind of a multiple choice one. any kind of antimicrobial agent you want people to take, you want it to make really inexpensive, you want it to act really fast, otherwise people are not going to use it. They'll say it takes weeks for this to work. You need to be able to store it, or people need to be able to store it, or they're not going to use it. And they want a big enough amount of it that they can purchase and then just store and then use it whenever they need. And then being able to control all these microbes that we want, all wrapped up into one. So it's pretty heavy requirement for the pharmaceutical company to come up with all of these. Yes?
When you use an antimicrobial agent that specifically targets like proteins or ribosomes, does that antimicrobial agent need to be partnered with another that can break into the cell wall? Because how would those agents get into the cell?
Well, usually the reagent is small enough that it can go through and can get into the cell. Yeah, because it would be a great idea if we could partner two things together, but then that's going to make it even bigger to have something that gets to the cell wall and maybe is tagged to something else and say, okay, you go through, I broke it open now, and you can go through this, because then everything else could. So I don't know of any that are like that. Maybe it could be a combined a molecule that they make, and that could make an incision and then also send the whole molecule through. But yeah, it is difficult to get things inside the cell if we're trying to penetrate that. Again, these are things I think you can just kind of read them. I don't need to go through and do each of these for you. So efficacy is really important. We just want to make sure it's going to work. And we'll do that. And then prions, these are things that are really most resistant. And these are most susceptible. So prions, have you heard of prions before?
Yeah.
Okay, so probably most of you have. It's pretty, unique. You figured, 25 years ago or something when they were first brought up, I mean, it was poo-poo. There was no such thing as these misfolding proteins, etc. But now we know there are in more cases. But anyway, these are very resistant, and then down here, these envelope viruses are much more susceptible. So again, nothing that you would have to look at this list and then be able to name which is the highest and which is the lowest. Nothing from that list. And here, again, these factors that are going to affect it, of course, all our environmental conditions, our temperature, our pH, all of those things are going to be something that could be affected. And this organic material, organic material we have to watch because it is organic. It can be denatured, heat can harm it. There's lots of things, so you have to really watch when you're using organic materials. And so these kind of things, this is just saying, the higher the temperature, the faster the organisms are going to die. Looking at graphs, it's really important that you look and see what's on each axis of the graph. Here's time in minutes, and here's the organism. Sometimes if you misread a graph, you can totally misunderstand what it's telling us. But you can see here is the temperature, and in time-wise, this temperature means that it's going to kill it much quicker than something that has a higher temperature, and that's all this graph is doing. So these are the biosafety levels that you should know. So these are important. I think they're telling us our next lab that we do is biosafety level 2. Maybe it's the lab after that, but there's one lab that they mentioned that. So these are something that you should know. Okay. So this is a good slide. This slide and the very first slide kind of are really good slides to know. And this also is in our, this already was, this was already in our lab manual. So I think it's either this lab or maybe the next lab where they actually give you all this information again. So you know it's going to be important. Okay, and then this one, it says why are biosafety level 4 suits pressurized and why not just wear tough regular suits. So this one is, these are pressurized so that the pressure goes out. So they're in the puncture, nothing that's in the environment is going to go into. It's all the pressure is coming out of the suit like this, pushing everything away. So the pressure is going out this way. So if anything's out here, it's not going to be sucked in. It's always going to be pushed away. So that's why those suits are all pressurized.
You're saying that's something we need to remember, or we're gonna?
Well, I mean, I think as curiosity, you would say, well, that's an interesting fact, so.
Like pressurized going outward?
Yes, yes, because you can see the suits are puffy,
Yeah.
so the pressure's going in and then forcing it out. So if they got a hole in it, then it would be like the balloon is going shh, everything's going out. So it wouldn't be sucking in, and that's what you wouldn't want. We wouldn't want all the microorganisms or whatever that might be in that environment being sucked into the suit with you in it. So yeah, just kind of an interesting fact.
Like a COVID lab or something like that?
Yeah, I know this is Ebola, I think.
Ebola?
Yeah, Ebola. Yeah, it's just such a horrible disease.
Wow.
Okay, and these two, I think they were in our lab exercise this last time, weren't they? I think in your pre-lab questions.
Yeah.
Okay, so you can see how the lectures, we should have had this lecture before, and so that would have, then we'd had information in the lab, and you would have tied those two together. So these two things that you should know only because they're also in the lab manual, and they're here. And moist heat and dry heat, we can understand that moist heat might be much better than dry heat. So to incinerate something is really good, but this moist heat can be brought to a higher temperature. You think of the autoclave. So the autoclave will have an extremely high temperature because it's pressurized, so the water can't boil. So they can turn up the heat higher and higher and higher and higher, and the more pressure that they have, the water still can't boil. And all of a sudden, when it starts to boil, it's going to boil at an extremely high temperature. It's not 100 degrees like water normally boils. It's going to be much, much higher. So the autoclave is based. The autoclave is based on that high heat. Okay, and I think there's only a little bit more on here I even wanted to mention. So pasteurization, you should read this so that you understand a little bit about pasteurization. Here's our dry heat that we use, and this gets extremely hot. And then here's our Bunsen burner. And so we're just kind of thinking about the microbial growth. We have different levels that microbes grow, and we already had that in our last lab manual where we had the different graphs. Things that grow at different temperatures. And this term, lyophilization, this is when you have freeze-dried things. So that's just a way to store things long term. So this is a good term. This means that if you have to store them for a long term, you can take all the moisture out of them and freeze-dry them. And we don't do this in this lab, but this is a way to sterilize media. So you put some non-sterile media up there, and you have these membranes, and the media has to drop through the membranes, and then this would be sterile. The membranes have little tiny holes in them. These aren't the holes, these are organisms, but they have little tiny holes in them, and you can buy them. They're very expensive, that they have itty bitty holes, and if they're a little bit larger holes, then it's not quite as expensive. And that's because what they're trying to do is eliminate different organisms from getting into the sterile environment. So depending on the size of the organism, either bacteria or yeast or even viruses, the holes will get smaller and smaller and smaller so that they can eliminate them. Okay, and that's all this is a way to filter, sterilize something. You don't have to know anything about these size of pores. You can just see that as you get smaller, 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 you can even take viruses out of a solution. But that's not important, just so that you know that the size of the pore will be dictated by what type of organism that you're trying to eliminate from your sterile media. And this is the, this is a regular hood. And you can see that the hood, we have one like this in lab. It's not quite as fancy, but you can see the air is going from inside and then up and out. So the air is not coming out. It's all sucking everything in and then making sure that whatever's inside there is going to stay inside the hood. And that's why it's important for us when we're working near it that you keep it as low as possible and not let any of that air come out. And hypertonic, hypotonic, I don't know if we've gone over those terms before. So we have. We talked about those, okay, so here, this is the idea that if you have something that's hypotonic, that means the region around the cell is not going to have very many particles, and so the water inside the cell will come out. Okay, so if something is hypotonic, there's not very many particles, there's not very many particles, and then the water will go in. And so here, the hypertonic is where you have a whole bunch of particles, and the water molecules will come out of the cell. So here, hypertonic is where we lose the water from the cell, and hypotonic would be where the environment will take the water and put it into the cell and burst the cell. Okay. Radiation, we will have a lab at the very end of the semester where we will take vegetables and we will take fruits, and then we will see how much bacteria are on them. So we'll take them and spread them on media, and then we'll make decisions on how many bacteria we think are on them. And all of them have been ionized for the radiation. Here's some strawberries. They have this little symbol here. I shop at Trader Joe's a lot, so I went to Trader Joe's and I looked all over for the symbol. They don't have this symbol. So they don't buy any fruit that's been irradiated. I guess there are people who shop there, for some reason, do not feel that that's appropriate, and so they don't. But at most grocery stores I went to, I could see that they had the symbol on the fruits and stuff that they were selling in packages. Okay, but this just means that organisms are on there, and the longer you leave them, they're going to start growing. So there's just no way to stop it. Non-ionized radiation, this is really things that like UV light, which we are going to use next lab, I believe it is. We'll put our stuff in the UV hood, and in that UV hood, we'll have a cardboard like this over our plate. So we have a plate like this, let's say, and we put it on half of it. So this part will be protected from the UV light because it's a paper, and this part will not. So this part, you'll see the organisms die, and this part, you'll see them live. So it's pretty amazing how UV light can kill organisms relatively soon. And we work in groups, and we have some people that take 30 seconds, somebody will radiate for like 45 seconds, somebody will do after 1 minute, et cetera, et cetera. And so we'll see the gradations on how that affects microorganisms. Okay. All right. I don't think there's much more on here that none of these structures do you have to know. You don't have to know about phenols. Alcohol, this is what we use in labs, so alcohol is what we wipe the benches and stuff with, so you should know not what it looks like, but you should know about the swabbing and stuff of the 70% ethanol. And so one thing I would like to mention, if you swab something with 100% alcohol, let's say, versus 70%, which one would be more effective? 100%? So why would that be? More powerful, more potent. More potent. Okay, so why do we use 70%?
Because it's the danger of...
Pardon me?
It's safer.
It really doesn't have anything to do with safety.
Less expensive?
Oh, I'm going to sneeze.
It's toxicity, right? I mean,
So, no, this is, when you put ethanol on there and it's 100%, it'll dry almost immediately.
no,
So if you are a stack of bacteria, which bacteria are not alone on there, here's a stack of bacteria. If you put the ethanol on, these few die off. These guys underneath live. But if you put 70% on, it sits there for about, what would you say, at least a minute and a half on your bench?
Yeah.
You put it on and it's still wet and you can't put anything on it, or you can smooth it out. So even that lingering amount of water is going to seep through down all into the bacteria, and even the bottom layer of bacteria will be killed. So 70% is much more effective than 100%. Okay, so that is something that you should know, because that's what we use on our benches. Okay? All right. Halogens, I don't want you to know anything about those. Oxidizing agents, hydrogen peroxide, we are going to use that we didn't use it on this one. I think for the catalase, you'll be using catalase, and I think you'll be using this hydrogen peroxide. So this is for catalase, and catalase neutralizes that hydrogen peroxide from the oxygen radicals. So hydrogen peroxide I would want you to know only because it's going to be involved in one of the tests that we do. But other oxidizing agents aren't really important for us, but this one is going to be important. And these are surfactants, and so just the word means it's going to be a kind of a detergent-like molecule. And these surfactants have these kind of tails like this. These are all hydrophobic tails. So if we have a membrane, like this kind of membrane, that hydrophobic molecule can go right through the membrane, because this is also hydrophobic. So this surfactant can actually dig into the membrane and then start separating the membrane. So that's how soaps will work. So detergents and soaps have these hydrophobic, and then the hydrophilic part is this part that's up here. That's got a charge and stuff to it. But nothing that you would have to draw, nothing that you would have to recognize. But you just know that surfactants are things that are going to interrupt the cell membrane. Okay, so none of this stuff you need to know. Aldehydes, you already know what an aldehyde looks like. But I wouldn't expect you to look at formalin and know what that is. Okay, so you don't have to do that. Just aldehyde, you already know what an aldehyde looks like. And all these, you know all these. This is only important because this is how a lot of things that can be damaged with heat, etc. You can use gases or substances like when we had anthrax and we had mail services. And they have all those machines and all those small little parts. They had to put gas in because gas can seep in everywhere to kill the anthrax. But if you tried a spray, if you tried anything else, it would not be able to get in every crack and crevice of all these mail houses that were affected by the anthrax. Okay, and so ethylene oxide, this is the one that's I think they did use for the anthrax. You don't need to know any of these names. This would be the only one name I think would be good to know because at least you know about anthrax. And enzymes that we have, these are enzymes that are going to reduce bacteria and preenzymes. You don't need to know this one. They use that on medical instruments, but we don't really deal with prions. But lysozyme is a good one, and this is ones that we have in tears. And also other bacteria have lysozyme with them, and they can actually kill another bacteria with lysozyme. So this one, human tears, we have it in, and it disrupts the peptidoglycan layer. So that's how it can kill a bacteria, it will get into between that peptidoglycan layer and be able to open up the bacterial cell. So when your eyes are tearing and watering and stuff like that, that's good. It's trying to kill bacteria or get anything out from the eye. It's not to drown the bacteria, but it's to actually disrupt the membrane, or disrupt the peptidoglycan layer. And we're not going to talk about these individually. We'll talk about the antibiotics much more later. And semi-synthetic just means you take an antibiotic that we already have and we try to just make it better. You know, we can't find any more antibiotics, so we just take the same antibiotics that we have and we just try to make them better. And so that's what this semi-synthetic is. And the synthetic ones are where we just we mimic whatever we have and we just make a totally new one, thinking that we can do something better. And this phenol coefficient, you don't need to know about that. That's usually how we judge how well things work. And we did things that we washed our hands with and our different antimicrobials and agents, and then we looked to see how they worked compared to each other. But usually you compare them to a standard, and this phenol coefficient is a standard that would be compared to. All you need to know is that this is a standard, but you don't have to know what it is. And then these other tests, we're not going to talk about them at all. And again, this test we're not going to talk about at all. In-use test, we are not going to talk about at all. Zone of inhibition, we're going to do the Kirby-Bauer test much later in the course. And this, in fact, we're doing a little bit of this now. You just put discs on to your plate, and you're going to see whether they have these halos around them. And this just means that the disc is secreting something from it that is harming the bacteria that you have on your plate. Okay, so these are zone of inhibition. So Kirby-Bauer, we'll talk about that when we get to it. Oh. And so this is just a little bit about antibiotics and antibiotic resistance. And it really lets us know what the difference is between maybe resistant bacteria and then when it's hit with antibiotics and then what grows. So when we have mutations, organisms are mutating all the time. I mean, our body mutates, but we have lots of mechanisms within the body that will either fix those mutations or just remove that from the DNA. So this is where a particular bacteria has already, before it got in to an antibiotic situation, it already is resistant to the antibiotic. It made mutations long, long ago, and it just can grow equally as well as the normal bacteria, but it carries this antibiotic resistance mechanism within it. So when it ever encounters an antibiotic, like in this particular situation, you take antibiotics, all the other bacteria are susceptible to it, but these ones are not. They are already producing an antibiotic resistance gene, and when the antibiotic is already on them, and so they can survive. It is not, it's a myth that just because you take antibiotics, the actual bacteria can mutate because of the antibiotic. They are always mutating, and they're going to find a particular time when whatever mutation they just made happens to be resistant to that antibiotic. And then you see this, they really grow. And the ones that aren't resistant are going to be far and few between. So now, whatever you had for this natural selection, so now we have a whole population that's going to be resistant to whatever bacteria that we had. Yes?
So when you cut your antibiotics in half, you're not allowing that bacteria to become resistant to it?
What you're doing is if you have, let's say,
so if they said take it for 7 days straight, twice a day, but you said I'm going to take it for 3 days, one time a day.
So why do you take it for 7 days anyway? Why are we taking it for that many days? Why not 11 days or 92 days or something? Why do we take at a certain time?
What the doctor ordered.
Yeah. Yeah, what do you think?
To avoid resistance to those?
To avoid resistance, but also, and it's not only those bacteria, it could be any bacteria. But remember the curve, and maybe it's the next lecture that we're going to have. There's a curve that goes up here. There's a lag phase, and the exponential growth phase, and then stationary phase, and then it dies off. There's that curve for bacteria.
Yeah.
In this point in time here, this is when they're making all their enzymes, etc. And this is where you have to kill the bacteria. So you have to have the antibiotic that's going to act on whatever the antibiotic is supposed to act on the bacteria when the bacteria is active. If it's up here and it's already got its cell wall made and everything, and you have an antibiotic that's going to attack the cell wall, it's not going to hurt those bacteria up there because the cell wall is already done. It's right here where they're trying to make their cell wall that you can harm them. So you take them 7 days, so every time they're multiplying, you kill these off. And then they start to multiply again, and you kill them off. You multiply again, you kill them off. And pretty soon after 7 days, there are no more bacteria that you can kill off with the antibiotic, and then you're done. But you're right, if you cut it in half, or you take it at irregular times, you allow the bacteria to go through this exponential growth phase, and they can overpower that antibiotic. And again, the antibiotic doesn't force them to make mutations, it just allows bacteria to grow. And because they have tons of bacteria every time they grow, it's just a matter of chance that they may get a mutation that will make them immune to whatever antibiotic you're taking. Yeah, but it's not the antibiotic that makes them. It's, if the antibiotic allows them to grow, it just gives them more time to make a mutation. Yeah, and that's why we are so concerned about, let's say, COVID and some viruses, because the longer we let a COVID virus replicate in bodies, then the ones that viruses that get out, might have a different strain. But if you take, let's say, vaccines, and the virus may infect you, but you don't allow it to break out of your body because the vaccine has kept it at bay, then it can't multiply and divide and it can't make new variants. So that's why being vaccinated for everyone is so important, because it keeps new variants from ever being developed. So the more you can take either antibiotics for bacteria or vaccines for viruses, we can stop those things from replicating, and that would allow us to eventually kill them off. Okay, so this is just, we're going to talk about antibiotics a lot more later, but this is just what happens if you already have a mutation in your bacteria. They're just going to take over. They're going to take up all the food. Okay, so not everything in this chapter, but I'll reiterate again. This one is really good if you go back to this. This chart. Yep, this chart is really good. So it's almost like kind of the multiple choice kind of thing that you can do. And these slides are posted so you can see this on Canvas. So I'm going to suggest that maybe we take a quick break in between and then just start this next chapter. So we can take maybe a 10-minute break or something. And this is microbial genetics. So again, this isn't something really important, I think, for a lot of our class, but the genetics part will come in as we move along in different labs even. So we'll do like a 10-minute break, and I don't know what the exact time is right now. Looks like it's 7:08. 7:08? Okay, yes, 7:08, that makes sense. So 7:08, so, and we can do maybe to 7:18, and then come back. And if anybody has any questions about anything, I had one person I needed to talk to, but they're not here today. Oh, he is here, but I don't see him.
That's our going home early tonight.
Okay. All righty, thank you. You lasted an hour, that's good. That's the criteria. So where is this?
Are we going to finish chapter 7 today?
We won't finish it.
Okay.
Yeah. We'll just do a couple of highlights from it, and then you'll kind of be on your own. This one is, it goes in, the podcast that's on here is very, very detailed. And I just do not think for our class that we need that much detail. There's only one thing. Have you ever heard about the lac operon? Maybe yeah, really cool. So a couple things like that when we're dealing with organisms and we're really trying to understand how do they make this enzyme, or how do they do this, when you really realize what's inside the bacteria, what's happening. And also when we're looking at something like catalase. Catalase, when you put that drop of H2O2 on it and it starts bubbling, you go, what's it doing and how did that happen? And it's because if you are an oxygen-bearing organism, you are going to have enzymes that is going to allow you to detoxify anything that is got oxygen radicals in it. And that's what it's doing. It's kicking in an enzyme called catalase, and it's making those bubbles because it's trying to detoxify the oxygen. So the more we understand about that, the more you could design experiments to be able to try to harm bacteria. But I mean, no matter what experiment we do, it's going to prefer not, because they will figure out a way around it. They're really very, very fascinating. And we have to learn to live with them. I think that's the only thing that we can realize. Okay, so anything about lab or anything else that you might need to know? And so tomorrow's lab should be really interesting to kind of see what your end result is. These kind of labs I like because it has a purpose lab. I mean, the rest of them were teaching you technique, which is really important to know exactly how you do something. Because we will have a couple more skills tests coming up. So it's good to practice them. So is Michael here? I didn't see him. Oh, there you are.
The other guy?
Yes.
There's two
Yes,
guys.
yes.
So there's one, the one,
Oh, we can talk about it. So what is it that you need? Yeah.
which is the one I'm doing physiology. And then there's the one with all the little squares.
The goal is to find the organism. So I think there's like several, let me see, 10 points for sure if you don't get it. Another three if it takes you too long, which that would add on. And focusing, so that's 7 points. Yeah, so handling, that would not do it. Stage lowering, that would not do it. Yeah, something like that, because that's the whole goal.
Yeah.
So that's why if you need to retake it, the max you can get is 20 points.
Right, so,
And you can only retake it one time.
yeah, so that's tomorrow.
Mhm.
Tomorrow we're doing it, are we
I
gonna
feel good about
discuss
that.
even with
The
this
lecture.
noted? Are we
About
gonna make
reading
sure
the
that
book.
they stand up in between this time? Like guarantee it? That they're gonna stand up
Essentially.
beside?
And then with the.
No, because the person can clean it up.
Yeah, I'm saying, are they gonna make sure, are you gonna make sure everybody does it?
Well, I'm gonna try to ask them to do that, but I would not accept a slide from somebody that cleaned it and had my grade dependent on it. I would clean it again before I put it on the microscope.
Okay.
Yeah, no, I would not trust anybody. So I'm asking everybody, please clean them up because it will make it go faster.
Everything is so much.
Yeah, it is.
I can't just do without.
Yeah,
So.
yeah.
Are both graphs?
Yes. I mean, I think, what I saw most people after, you see how fast they were
No,
finding it on the
she's
stage for
gonna
that.
reschedule.
And I think what it was
She's
is,
gonna reschedule
they
the
were
one she was
really
talking
cleaning
about,
them before they
but
would
she
start.
wants to explain to.
Yeah, so it's only just about,
I think tomorrow the other one.
less than.
Yeah.
Oh no, I think
less than-
both of them
I
she's gonna
went
reschedule for next week.
No, tomorrow, the 13, 14 is
Yeah.
due.
Yeah, no,
But
I
the five
mean, I think it was
is
just, I
gonna be
don't know,
next
it's
week.
a strange thing, even,
With the
anyway,
CS
not
and
said,
all that.
what happened?
Tomorrow,
I said,
tomorrow, what's due tomorrow?
So the one that you,
The one that I have.
13, yeah,
But
tomorrow.
that's, I think even brought, you did the slide you brought, you was
I have to
something.
redo that one.
Yeah.
Oh, I see, you touched it, but, oh.
Yes.
I'm just, it kinda looks like it, but mine's messed up, so.
Yeah.
Following the video. It's gonna get that Excel here in the library to change this, and in the video, the lady shows you how to do it, but I couldn't find the little.
Yeah. Yeah.
Yeah, in the lab, I can basically, I can see that's the one she's using is like a different version, different computer version,
Yeah.
so we have, so. That's why it's not good. Sorry, he couldn't.
Yeah, so they have, the test is supposed to be for 3 minutes, which I thought was too short. But then I realized everybody was just zipping through it, so I said, oh, 3 minutes is something that's logical. So I still won't impose that, I'm still gonna do 5 because I think anybody who's nervous about the time, you find it soon, so you'll feel better about it instead of saying, oh my gosh, the time.
The way I did.
Yeah. Yeah, well, he gave me those 2 slides, and I think they were cleaned up pretty well before they were put away, but we at least have 2 slides now that we can. But he also told me that he shouldn't have given me that second slide. So he said, that's not what the test is about, you can't have a second slide. And I go, okay, but we did, and so we'll keep them.
They'll take one.
Okay, so are we pretty close to our time so that we can be done here like 8 o'clock?
That's 9:45. Let me see if that's good.
Okay, so I think it is, it's 17, so we have 1 more minute, but I'll just kind of get this ready. So everyone should have had biology, so a lot of this should be something that you've learned before, I mean, some of it, when I read it again, I said, oh my gosh, this is, now I remember it, but I didn't really remember it when I first looked at it. So I think that might be how it is for you. Some of the words will be, like the bold words, you'll know what it is, but maybe specific definitions or something will be maybe something that you haven't looked at it for a while. Again, also there are posted the slides from the individual, I need to learn her name to say it, she's gone really in depth on some of this stuff. I mean, a lot more as I was listening to it, I go, oh my gosh, you know, there's so much depth. So I would not, if something in here doesn't make sense, then I would definitely go back to those and look, because she's very thorough, but it's just so much more detailed than we need to know. We don't need to know. On the replication stuff, I cut a lot of stuff out just to say, here's how it begins, here's how it ends, here's a couple of the enzymes that are used. All right, so let's talk about this. So microbial genetics is really important to us because we need to know how their genome works. If we want to kill bacteria, if we want to use them for research, if we want to do anything else, we have to understand how their metabolism works and all their genetic genes that they have and how they're turned on and turned off. So that's really what this chapter is on, and it's going to culminate with us talking about the lactoperon gene. So genetics is just the inheritance. So we're just understanding how these organisms actually pass on their traits to their progeny. And the genome is all the traits that they have, so kind of the same definition that you've had. So it's all their genes, all their nucleotide sequences, etc. And again, here I would not expect you to even recognize any of these, other than you would recognize that they were nucleotides. Okay, so not that you would have to draw them, but that you would just recognize there are. I also would expect that you would recognize that these are hydrogen bonds between them, and that G and C have 3 hydrogen bonds, and A and T only have 2 hydrogen bonds. And that probably snaps something back in your memory, and you go, oh yeah, I remember that. G's and C's have 3 hydrogen bonds, and A's and T's only have 2 hydrogen bonds. And then this is phosphodiester bond between these. This is where the bond comes, so here's the backbone, and then these are the bases and the sugar, and so they're connecting together in the middle. So this is the alpha helices, so this is all the stuff that's in the middle here. So those are all hydrogen bonds. And we know those are relatively weak bonds so that we can rip it apart, copy one strand, and then put it back together again and not take much energy. And these are some of the things we're going to talk about: the structure, replication, function, mutations, repair. I'm not going to do too much on the repair. That's not, it's beyond what we really need to know. And I don't really want to contrast the bacteria with the eukaryote. That's probably what you learned in biology, is mainly about eukaryotes. But we really are talking about bacteria, so I'm going to emphasize those more. So that cuts out a lot of the slides. And so this is a lot on this slide, but I tried to put bullet points that will just make sense. So the nucleic acids, these are polymers of the nucleotides. So you have each nucleic acid, they're going to be put together, and that's what's going to make the polymer of our nucleotides. And then these hydrogen bonds, they're the ones that form between them. And of course, DNA, what it does is it stores all the information that we need to recreate, whether we create us or bacteria recreate themselves. The sugar, phosphate sugar backbone, that's what this is. So here's a sugar and here's a phosphate, so that's the sugar sugar phosphate backbone, and that's what the phosphodiester bond is. And these are read from 5'. They terminate at a 5' carbon, and here's a 3' end, it terminates at the 3' OH. And so you can see two different strands. One's a 3', one's a 5. Here's a 3, here's the 5 up here, and then this goes 3 to 5. So it's always read 5' to 3'. We'll see a couple slides that point that out. And if you looked at a strand going all the way through, like this is a 3', if you followed it all the way over here, it would end in a 5'. And then here's a 3' over there, and it's going to follow all the way, and it's going to end in a 5'. So when you look at them, you can see that they're going to be complementary. And there's hydrophilic faces, the outside. So the facing of this is all hydrophilic out here. And remember, this is all done in an aqueous environment, so it has to have something that can interface with water. And the hydrophobic part is all going to be in the middle. Some of these are hydrophilic also, but the hydrophobic part is all going to be towards the middle of the strand. RNA carries information, so the storage of the information is in the DNA, and the carrying of the information is with the RNA. So whenever the cell wants to make a new gene or do something, it has to undo this DNA, take a certain portion of that DNA that has a gene that they want. They have to express that gene. The way they express it is they have to take the DNA where the gene is encoded and make it into mRNA, so that it can, for us, it can leave our nucleus, but for bacteria, it can just go into the cytosol, and then the ribosome can go ahead and translate that into a protein. So we're going to look at that step by step. And of course, there's a lot of DNA. All of this is packed really tightly. This is kind of an amazing fact. 3 million micrometers go into 5 micrometer nucleus. So 3 million micrometers, I don't know how long even that would be, way around the room a couple of times. And that's going to go in the nucleus of a cell, 5 micrometer nucleus. So it's pretty amazing. And we have all kinds of like histones and things that wrap this up. We're not even going to worry about that. We just know that it has to be wrapped around something so to efficiently be fit into the cell. And here, mainly, that's what I want to focus on, is the prokaryotes. So they have chromosomes. They're distinct chromosomes. Sometimes there's one for a bacteria, sometimes there's a couple. There's a couple bacteria that have up to like 10 or so chromosomes, but mainly they're only 1 or 2. And it's usually a single copy, so they're haploid. They don't have, like we do, we have a diploid where we have 2 different copies of it. And they're generally circular, and they're in what they call a nucleoid. So why are they probably circular? I mean, why not just make them linear?
Travel?
Pardon me?
Travel?
Travel, so you mean when they're replicating it? I mean, travel is a great word. So when they're, I said they replicate all their genome and they totally divide every 20 minutes. So can you imagine trying to take a chromosome, you have to go here, and then you have to jump off, and you have to go to another chromosome. I mean, it could take forever. So they just go around in a circle and they're done. So now all the genes that they have to express, they're done. They can replicate their DNA, they're done. So that's really why they do it. It's so fast to do. And then this is just a picture of, this is a bacteria and it's been split open a little bit, and here's all the genomic material that's all spewed out from the cell. And we were going to do something called the string test. Last time I think a lot of you filled out all the stuff for it. And when I went in that morning, or that afternoon, I said, are we going to do that this time? We didn't do it last semester, and he said, no, we're not going to do it this time either. And it is where they kind of take some of the DNA from, I don't know what it is, like a strawberry or something like that, and they get a lot of DNA out of it. It's all sticky and stuff. It's kind of interesting to do, but we just didn't do it. So they're circular, and they're in a nucleoid. We have a nucleus that we put our DNA in, and you can usually see it in one area of the cell. Bacteria, they have, it's a simpler organism, supposedly, and what they have found that they don't have nucleus, they don't have membrane-bound organelles. What they have is proteins, and proteins will make little gates, like. And what they do is they find maybe this area might have some protein that surrounds it, and it makes like a nucleoid. And that way then the DNA stays there. Sometimes the DNA also is tethered to the side of the wall to keep it in a certain place. They don't really understand why it always, when they look at micrograms or microfilms of this, they see this exact same structure. There's always something right here and something right there. So it really looks like bacteria have some organization to the way they put things, and it's just duplicated in all species. All right, so circular, maybe you have 1 to 2 distinct ones. DNA has all the proteins so that we can package these up into RNA, and then those can be expressed with the ribosomes. Plasmids are another thing that they have. So even on this, they show here a little tiny plasmid. This little loopy looking thing here is a plasmid versus all of the genomic DNA. And they have plasmids because they get genes from other organisms. They can store it on a plasmid. When they are too busy to do anything, they only replicate their own genetic material, and they'll just throw out the plasmid. Because the plasmid does not have cells or have genes on it that they need in order to replicate themselves. So those little plasmids are just extra things. Some of them have many, and some of them have almost none. But those plasmids are things that we use in science to be able to put, let's say we can put human growth hormone. So we can put that into one of those plasmids, and then we can put it in the DNA, we can tell into the organism, and we can tell it, go ahead and start replicating this for us. And they can make a ton of the genetic material that we want. So human growth hormone, they secrete it into the vial that they're growing in, and then we just have to harvest it out of that vial. So they're very, very useful if we take a plasmid and we put something in it that we want from them. Okay, so these plasmids are very small, they're circular, carry information for their own replications, 1 or 2 other cellular traits. They're not essential for the normal metabolism, so the organism can throw it out and not be affected by it. It gives usually some survival benefits. Maybe it will have an antibiotic resistance gene on it, or it will have something else that's useful. Otherwise, then the organism is not going to keep it in. It's just too much trouble. So some of the things that can have on this plasmid is a fertility factor, a conjugation tube that will allow it to connect to other cells. Resistance factors, so this bactericin, this is a factor that's a protein toxin that can kill other bacteria. And then they have these virulence plasmids, just that they have virulence genes on there that make this organism more virulent. So those little plasmids are really important to some bacteria, but it's not essential for them. And this is nuclear and this is eukaryotic, so I'm just going to even kind of skip this. This is just showing all these histones and how you have to wrap the DNA all around it, package it up smaller and smaller and smaller, and so then we can get these chromosomes. But since we're not really going to be dealing with that, there's like 3 or 4 slides that have this information on it, and so you can really skip this. So it's not something we're really going to talk about. Okay, so again, here's eukaryotes, and this is some extra stuff that we have about eukaryotes. I'm not going to talk about that. Okay, this is eukaryotic stuff. All right, so this is kind of a helpful chart. This is eukaryotes, but again, we're not focusing on this, but it is interesting to see these are haploids and these are diploids and stuff. So it is an interesting chart, and I can see me asking, are plasmids in eukaryotes or only in prokaryotes? What's the difference between the nucleic acid, circular or linear? So I think this would be maybe just good to look over. Good for like a multiple choice kind of question. All right, so DNA replication. DNA replication takes energy. So where do we get the energy from? It's from the triphosphate. So I think I put one on here. So here is the triphosphate on a nucleotide. Usually we use ATP, but it's on this slide, so I thought I could use this. So you can see these three phosphate groups that are on there. Whenever you need some energy, you just cleave off one of them, and that gives the amount of energy that we need. So if you go back here, this triphosphate deoxyribonucleotide is the thing that's going to actually give us the energy and then give us a monomer that we can connect together. So each of these things are, as we replicate the DNA, we have to have these nucleic acid monomers to connect together to make the DNA or the RNA. Okay, and that's what we call replication. And the replication that we have in this is semi-conservative, meaning when we open this up at the origin and we start traveling around, one strand is going to be the daughter strand of the original, and one strand is going to be new. So you always have the old daughter strand and a new strand that you're making together to make a new piece of DNA. And these are the structures. Again, I told you do not have to recognize them. You just really need to know that G's and C's have 3 hydrogen bonds and A's and T's have 2. And that thymine and adenine and uracil will bind, but uracil and thymine are in RNA is uracil, and in DNA is thymine.
I'm sorry, can you repeat the G and C?
Yeah, so you can, it's hard to see, but this is guanine and this is cytosine, and you can see they have 3 hydrogen bonds. So they're really tight together, or tighter together than if you only have 2 hydrogen bonds. So A's and T's have 2, A's and T's have...
I see that now.
Yeah, these two only have 2 hydrogen bonds. Yeah, so it really makes a lot of difference, like when you're doing sequencing and things like that, but we won't do that until about 4 or 5 chapters from now. Okay, so this is a cartoon figure. So here's our phosphate backbone, here's our sugar, just a cartoon figure, and then here's all the nucleotides. And you can see that as we add another nucleotide on, let's say we want to add this one on, we are going to use one of its phosphates to add the energy to allow us to elongate our chain. So making DNA or RNA takes energy, and we get it from those nucleotides that are, or triphosphates that are added on. And again, this, it has some interesting things on it, and only the words that we have on here are the ones I'm going to ask you to remember. So here is our DNA. We want to actually replicate the DNA, so we want to divide that cell for some reason. We have to have this thing called helicase. You need to know that. So the helicase is going to come in, and it's going to help this start to separate. And then we have these stabilizing proteins so that they don't come back together. Otherwise, these things could kind of go back together. And then we have the DNA polymerase 3. So this is the DNA that's going to actually polymerase that's actually going to start making the new strand. And you can see it up here. It's going to be adding these nucleotides one at a time. So really, helicase is the initial process. The helicase has to come in, kind of spread it open. These stabilizing proteins come in to keep it from going back together. And then the DNA polymerase 3 just comes in and starts going. And again, catalyzes synthesis from 5' to 3' regions. All right, so the next one, way more than we really want to talk about. I think the previous slide that I gave you is good enough. I don't care if you know about the Okazaki fragments. You do need to know that we have a leading strand and a lagging strand. I think it's just nomenclature that you need to know. It's just that when you open this up, and we can only go 5' to 3', this is going in the opposite direction. So we can't go this way on this strand. We have to go the opposite way. So we start right here, and we go this way, and then we have to jump and then start again. So we have little start points, and that's what this Okazaki fragment is. Again, I just want you to know that there's a fragment that on the lagging strand that is going to be produced, and on the leading strand, you'll have the whole strand being produced at the same time. We need a primer to get this started. We have triphosphate nucleotides that we add on. We have a lagging strand, and we have a leading strand. And that's all you need to know. There's a little bit more information on here, but that is not that important. Okay, so this is just how we're going to replicate the genetic material. And all organisms basically do it exactly the same way. So you can skip this kind of slide. If you go to the regular slides and look at it, there's a little movie there that shows you what's going on. In fact, if you look at the slides that are posted from an instructor other than myself, they pull in a lot of the videos from the text. So they show in detail exactly how things replicate and stuff, but I just don't think it's going to be useful for us. We're not going to use it in lab. It's not something other than if you've never had any kind of biology class, then I think this would be good. But to me, this is microbiology. You should have already had your biology someplace else. And now this is just how does this apply to prokaryotes and what we're doing in lab. So I'm skipping a lot of the stuff that I don't think really is relevant. And you can always go back and look at it. Okay, so here's a clicker question we can do. So which of these steps is first in the order of events in DNA replication? Oh, you forgot your clicker thing? Yeah, here's our clickers up here.
Oh.
You're already answering. Okay. So I'm gonna go back so you look at it. Ah, good choice. So I'm gonna go back and look at this. So here we have, we can see number one right here. So this little primer is added, and then a couple of other things happen, and then only down here are we getting to our helicase. So our helicase is going to be here. It's going to keep this fork, but in order for the replication to happen, we have to have this primase. So really, there's two answers that I can see. The primase has to lay down the primer in order to start, but the helicase has to unwrap it so that we could actually keep going. So this is a good answer. This was the answer that they wanted to have. It has to have a primase, otherwise the polymerase can't even jump on it. Okay? And then this is the last stage that we have. Okay? All right. And then there's this, oh, I thought I had another one in here. Okay, so I didn't. Let me see. Okay, I didn't, this one. Nope, okay. All right, so the replication, eukaryotic cells, we're not that interested, so we're not really going to talk about this. This is just a little summary of it, so we won't deal with that. All right, so genotype and phenotype, those are important words that you know the difference of. Which one of the words tells us about what we see in a person? The phenotype, yeah. So when the gene is expressed, it does something, and it produces something that you can see. And that's kind of what happens when we have our organism, and let's say you have catalase. you are going to put some drops of hydrogen peroxide on there, and you're going to see, is there an expression of catalase from the organism? So you're kind of seeing the phenotype of the organism because it's going to be expressing, or they're going to have bubbles, and that means that catalase was expressed by that organism. Because we can't see what's happening. So the only thing we can see is by a physical either outcome in color or size or shape or something. That's the only thing that tells us anything about the organism. So genotype is the genes, and phenotype is a physical thing that it does or the function that it provides for the organism. So two really important terms. And then maybe that could even be definition terms, that short answer kind of terms, because those two are really important. And then the transfer of this genetic material, we know it's in the DNA, that's where the genes are, and so now we have to get that expressed so that we can see a phenotype from it. So we have to go to, through transcription. The information is encoded on the DNA, and we have to, the DNA is usually very precious, so we don't want to use the DNA for anything except making mRNA. So we transcribe the DNA into mRNA, and mRNA then is something that, if it's a eukaryote, it goes outside the nucleus and goes to the ribosome. If you're a prokaryote, you just go from the nucleoid right into the cytoplasm, and the ribosomes will actually take that mRNA and then try making it into the protein that it's encoded for. So DNA is transcribed to RNA, and RNA is translated to these polypeptides or to genes. And gene function, this is kind of what our DNA looks like. So this is our genotype. So when we transcribe it into something, we transcribe it into mRNA, and then that is going to be translated by ribosomes. And so this is just to kind of reiterate that point. And again, if you look at the material that's posted, they have a whole bunch of videos that go back to things. There must be 15 videos in the stuff that's posted. So if you're really interested, you can. But if you just look at these slides, this is pretty much what I want you to be able to understand. So this would be an important thing that we're going to say about 5 different times. Okay, so if we go back here, so there for RNA transcription from DNA, because we have the DNA and then we have to transcribe that into mRNA, but there's more than just mRNA. So mRNA is really short for messenger RNA. So it's taking the gene or whatever it is that was designated to be expressed at that time, and it's bringing it out intact. And you also have things that come from RNA, or come from the DNA that goes into RNA, is RNA primers. So we could see on that one strand that you have to prime the strand with an RNA primer in order for the DNA to bind it, in order to, the DNA polymerase, in order to bind it. And then you have rRNA. So this is called ribosomal RNA. So we have ribosomes, and they look like this. Here's a ribosome, here's the small subunit, here's the big subunit, but usually it's drawn the other way around. Let me make this big subunit up here. Here's the big subunit, and here's the small subunit. This is a ribosome. This is the mRNA that's being brought through. But this ribosome is made up of a lot of RNA. So RNA is the material that makes up this shape of this ribosome. So that is what the rRNA is. And then in order to bring each of these nucleotides in here, we have another thing that's called a transfer RNA. So it transfers the right codon to the right place. Okay, so all of these are parts of RNA that comes from DNA, it's encoded on the DNA. And this is, there's several other slides after here, but I think I got rid of them. Okay, I'm going to flash past that because I consolidated into this. This is all I want you to look at. So now that we have our, there's a promoter here that says we want certain genes to be expressed. So from here to here, we want to make this gene. So there's a promoter in front. We have a transcription factor, like a clothespin, that binds onto this and says, okay, here's the point. This is the gene I want. And then this is where the RNA polymerase will go to the DNA and only transcribe that particular gene. Because we don't want the whole genome, we just want only specific genes. Okay, so, and this is going to have to occur in several stages. You have to initiate it, then you have to elongate it like this, and then you have to terminate it. So when you terminate it, there's this little rho factor over here. You don't need to know that, but there's a little factor at the end that kind of pushes it off. It says, okay, this is the end. Now we know where we start because of the promoter, and we know where we end because of this rho factor. Okay, so these are the 3 steps. And these slides that I meant to get rid of, this is the whole thing in detail. So I'm not, I just think this is more than we need to really go into. So I have eliminated that. So if you wanted to look at all this, this would all be on there. Okay, so I'm not expecting you to look at that, just that first one that I showed. Okay, does that make sense? That's enough information, I think, and you can always go back and look at those a little bit more. Okay, in eukaryotes, this is a little bit different. Again, on my slides, I have gotten rid of this so that you didn't even have to see it. So again, we're not going to talk about the comparison between the eukaryotes. Okay. And you've probably all heard about capping and polyadenylation and all that. This is not something that we're going to even be talking about with our bacteria, so I'm not talking about it now. All right, and so this, and here's what we talked about before. There's some promoter. Genes have to be located. You know, you've got a big long piece of DNA, they've got to be located somewhere. So they have these little promoters in front, and again, like a little clothespin type of molecule or a protein binds right here, and it kind of indicates here's the gene that you want to replicate. So everyone's going to have a promoter so that you get the right one replicated. And then you have to have a start codon to start that. Again, you don't need to know what the start codons are, but you need to know that you're going to have to start it somewhere, and then you have to stop it from somewhere. So you start and you stop, and there are various codons for that. Again, we don't need to know exactly what they are. So again, we're going from 5' to 3'. Always think of that, 5' to 3'. And these are ribosome binding sites that the ribosome is going to have to figure out when this piece of mRNA is in the cytoplasm. They go, oh, what is this? And it finds this, and they go, oh, I can bind right here. This is a site for me to bind to. So they're recognizing the mRNA by this kind of a site that tells them the ribosome should start here. And then the start codon just confirms that. Okay, so now we're going to make a polypeptide, which would be essentially a protein. And it's really done the same way. Introns and exons, this is what a eukaryotic cell would have. And the introns are the things that stay in, and the exons are things that we keep, and then the introns are things that we don't have. And unfortunately, a lot of mutations that are made can be made in all these things that we don't even use in our genome. So we're probably getting a lot more mutations than we know. So again, transcription is going from DNA to RNA. And then from here, we're going to translation. We're actually making a protein for that. This is what our ribosome looks like. I made a ribosome right here. This is what our ribosome would look like. And so here's our RNA. And I said that we have this rRNA, which is the ribosomal RNA. So you can see all the red of this. This is all folded up to make proteins. And this is our a subunit, our 50S subunit, and here's our 30S. This would be our smaller one. This would be our bigger one. And this is how they look together. So we also have one that's made up of rRNA, and this is a 60S and a 40S, and this comes up to an 80S. This is what eukaryotes have, a little bit bigger ribosome, and this is prokaryotes. So the beauty of this, yes?
Why don't the subunits add to what we labeled in there, like 50 and 30? Don't add up to 70 and like 60 and 40
Yeah.
to 80. Why are they like that?
Okay, so this means Swedberg. So it's a centrifugation. So when they put these units in, they centrifuge it, and this went down to 30 Swedbergs, and this one centrifuged down to 50 Swedbergs. That's probably the opposite, right, because it's heavier. And so when they added them up, when they put them all together and kept them together, they only went to 70S. So they don't just add these two together, they say all together that would be 70 Swedbergs where they would centrifuge to. but separately they would be 30 and 50. Yeah, it's really weird. Everybody always asks that. And the same thing here, these don't add up to the right thing. So one of these things that we will talk about when we talk about antibiotics is antibiotics can target the small or large ribosome. So when we target these, you can see that these are really different than these. These are eukaryotic and these are prokaryotic. So that means that we can have an antibiotic, we can take an antibiotic and it's not going to harm us because our ribosomal RNAs are different. So they're actually structurally different. Antibiotic is not going to bother them. However, we have in our mitochondria, which probably are descended from bacteria, have the 70S ribosome. So when you are taking certain antibiotics, and they usually do not allow younger children to take certain antibiotics because they are still developing many things that are the genetic material for that development comes from the mitochondria. So if we target the 70S because we think we're targeting bacteria, we will be targeting our own 70S ribosome in our mitochondria. So usually young children are the ones that are still developing. Older adults and stuff, those genes are already in place. We don't have to worry about them. But anyway, that's always a good thing to keep in mind, and this will be something that I definitely will bring up at a later time. So that's good to know. Okay, then I think. Right. All right, so these are codons. Again, nothing that you have to know, but just know there is a methodology that all these codons you can easily find what they would be. So codon is nothing except 3 different letters that are going to allow a specific amino acid to come to that site and be put into the protein. Okay, and everything is going to be codon to this. It's not going to be a UC, it's going to be a TGT when it binds to it. Or an A, sorry, because U's and T's are the same. All right, so this is the transfer RNA that's going to come. Again, you don't need to know the structure. This is really what they show most often. This is the anticodon that's on the bottom. It's complementary to what they're going to see, and it just would bind in. So this is what part of this transfer RNA is some of the RNA that is in our DNA that's transcribed into tRNA, mRNA, rRNA, etc. All right, so here's what the thing looks like. Hurry up, it says. Okay, let me go back here, and I wonder what happened. All right, I'm not going to talk about any of these mutations. I sat at my computer when I was back at Fullerton, and I deleted a whole bunch of these because there's just not stuff I think that we need to talk about. So let me go back. I want to talk about that one operon, and that's it of this part. Okay, so I got rid of a lot of this. The idea when you're doing translation, and this is coming from our ribosomes. Okay, here's where we left off. It's coming from our ribosome, so here's our big subunit and our small subunit. So in here, embedded in here, we have these sites. You probably remember this from other biology. And this is where the amino acid comes in, then it goes to the P site, and then it goes to the empty site where it actually leaves. So that's really almost the only thing. I have one other slide in this that I'm going to talk about, and the rest we'll just go through quickly. So here's our stages: initiation, elongation, and termination. You can see here's our initiator. It's a FMET. You don't have to know that, but there's an initiator one that comes in, kind of like a start codon. And we use energy. We use the phosphate from a GTP, and it goes to a GDP. So now we have the initiator started here, and now we're ready to go. So our next step is Then up here, you can see another, here's phenylalanine. This is going to come in. This is going to be our first amino acid of this polypeptide. This one comes in, everything hops over a little bit, and now we have the polypeptide getting longer. Here's the primer site, and then here's our phenylalanine. And then we have a threonine that's going to come in, and you can see it adds, and then we have a whole bunch of amino acids, one at a time they're added. So this is going to make our protein, and our protein is going to start folding instantly because it's going to go into an aqueous environment, to a water environment, and it's going to fold as fast as it can. So this is all I want you to know. Just kind of see the visual that you start at the A site, go to the P, and then the E site is the empty site. Okay, not this at all. This is not really important. Oh, well, I guess this is important to the fact that on a strand of mRNA, you can have a whole bunch of these ribosomes all at once binding onto that strand. And what that allows is that you can get a whole bunch of the protein that is encoded in this mRNA. You can get a whole bunch of them started all at once. So a prokaryote can quickly make the proteins that it needs. So they just all jump on at the same time. And you can actually see it. It's really kind of a big complex. Okay, and then of course there's something that's got to terminate it. There's a little tRNA at the end that just says this is the final, go ahead and fall off, and so that we have our peptide is released. And 75% of the genes, they're expressed at all times, which is really kind of surprising. But there's a lot of regulatory methodology to this to make sure that the ones that don't need to be expressed would not be. Otherwise, you're using too much energy. And remember, the energy that's used comes from what? Phosphate, right? A phosphate group. Okay, so every one of these has some kind of a phosphate group that is adding in. Okay, so this is the last thing I want to talk about, and I just think that these are kind of cool. These are called operons. We don't have them in eukaryotes, that we have them in protein prokaryotes that need to regulate and use their environment quickly. So here is an operon, and this operon has this. This is going to be lactose operon, or lac operon, they call it. And this regulates our lactose catabolism. And lactose is made up of glucose and galactose. And whenever a cell, most cells would prefer glucose. It's a quicker molecule for them to use. So if glucose is available, it's going to turn off this gene. They don't want this lactose operon operating. So it would be turned off. But the minute there's something called cyclic AMP, cyclic AMP is an indicator that the cell is starting to starve. There's not enough energy. And so cyclic AMP, we looked at these like ATP, and this would be a triphosphate, so you'd have an adenine, and you'd have a phosphate, and a phosphate, and a phosphate. So when you have APP, so ADP, this means you have a lot of energy. I'm sorry, ATP, three of them. So when you have ADP, that means you only have two, you have a diet. So that means now you're getting a little bit low on energy. And when you have AMP, that means you're really low on energy. You've used up almost all the phosphates on your energy molecules, and so you're getting low on energy. And cyclic AMP, only mono, cyclic, this will cyclize on itself. That's an indication in the cell that it's starving. It needs energy. So in this particular case, and I think it's the next slide that shows it a little bit more, in this one, this regulatory region, you will have the polymerase, the RNA polymerase come right here, and then there's going to be a operon repressor that's going to stop it from moving. So until we know that we actually need to have a lactose available, and if there's no lactose in the cell, then this repressor is going to be here to say, don't start the gene because there's not even lactose around. Even though we have a signal, this cap, where is cap? Cap is going to be right here. This cap protein will come here and say, let's start. And it won't start because we don't have the lactose in the cell yet, even though the cyclic AMP says we're starting to starve. So we have to have two things happen. We have to have the cap here saying that we're starving, and we have to have the other protein that tells us that the lactose is available in the cell. And then it will make this operon operate. And the operate, what it's going to do is the operon has 3 different proteins. It's all the proteins that we need to take lactose in, get it in from the outside, get it into the cell, and then break it into 2 so that we have glucose and the lactose to use, and then start the enzymes ready that are going to start metabolizing the glucose and the lactose. Okay, so it has just several different genes that are all together. They're on one, there's like 1, 2, and 3. It's just a very, very fast thing. You need one of them, and then all of them are expressed all at the same time. So you don't have to express one at a time. Okay, so these operons, they're really popular and abundant in bacteria because bacteria have lots of cases where they want many different genes to be activated at the same time, so they have these operons. And here is that cap protein that I had mentioned before. Here's our promoter. So I think this is a better slide than that other one that kind of shows you. Here's our cyclic AMP. So this binds here on this cap protein to say, hey, we're starving, let's get going. And then we have this other repressor molecule that says, well, wait, we don't even have lactose here yet. So we're not going to express these genes. But the minute the lactose comes in, then this actually removes the repression, and then it goes and reads these genes. Okay, so I thought this was really good. So this I would like you to know something about. So this one I think is pretty good. And yeah, a lot more detail here. So if you wanted to follow up, you can look at all that detail. And I think, so this, when I had gotten rid of all these slides, so I don't want you to know this one. And I believe it's all the rest of these slides are not going to be something that we need. So I'm not going to talk about the rest of these, silencing the RNA. So that was the last thing. All these mutations we're not going to talk about either. Yes?
So the lac operon is only present in prokaryotic cells?
Yes, yes. No. In fact, there's really no operons at all, even in eukaryotic cells. So we don't use them. Bacteria do a lot.
Is it because eukaryotic cells are usually not in like harsher conditions where like they're going to be starving?
Well, we do. We have lots of starving situations, but there are so many genes that would be regulated differently. One signal, like cyclic AMP for us, would have this gene operated, that one, and that one, for every different reason. So I think there's no very common thing that happens. That they have a limited environment, so either they have it or they don't, where we can have a lot of alternatives to, let's say, sugar. So we don't want to have one pathway that has 3 different genes to operate an activity. It just wouldn't be used very much, I think. Yeah, so operons are really good, and they're usually only in eukaryotic cells. So there's a lot of other things here. I almost positive I had taken these out because I don't even want to go through any of the mutations. We will talk about these when they come up. Okay? So all of this that's on here, I will make sure again. I think in your stuff you don't have any of these. I took them out. Can you see in yours, in student ones?
Just like 2 different PDFs, right?
Yeah. So I made sure I took them out, and I thought I took them out here too, but I didn't. All right, so that's really all of this chapter that I want to talk about. And now, I mean, somebody was here a little bit earlier, and we were talking about the quiz, that's going to be on either Wednesday or Thursday of next week, and how many chapters that will be on. I believe the quizzes are all exactly 20 points. That's kind of the boundary that they have. And so it's chapters up from last chapter up into this chapter. So we will consider 7 and 9 as chapters that we have now covered. So if the quiz is on, it's probably Wednesday would be lecture quiz. Yeah. So Wednesday would be a lecture quiz. It would be then through 9, and then we'll have whatever new lecture quizzes that we have. Okay, and it would go from what we had from last time, and I forget where we left off. We had like 1, 2, 3. Maybe is that where we were on the lecture quizzes, or maybe a little bit more. Wherever we left off, The study guide, from last study guide, it will say this covers, chapters blah, blah, blah. So this one will start where that left it off, and it'll go clear up until 7 and 9. We did not cover 8, I don't believe. So the text skips that, and we didn't cover 5. I think that was one that isn't covered. Does that make sense now?
No, we did cover it.
We did cover 5, so there was one that they skipped.
Four.
Did they skip 4? Okay, so we went 1, 2, 3, and then maybe we went to 5, and then I think we had 6, and 7, and 9, and we didn't have 8.
So 5, 6, 7, and 9.
Well, I'm not positive. I would have to go back and look at each of them.
5, 6, 7, and 9.
Yeah. So again, I'm going to try to do the 2 study guides on tomorrow, which is Thursday. And again, it's 20 points. And really, I think it was like 25 last time. We had 5 extra points on it. It'd be the same kind of format. But now there's just a lot more chapters. So it makes it a little bit different, because harder because of more chapters, but it makes it easier in a way because it's going to be more general. I can't get into too much detail. There's only a couple of things here that I maybe went into a little bit more detail on, but most of it's very general. Okay, so I think it's going to be very similar to the last one that you had. And then I'll have to look at the lab one. The lab ones you're going to find that are going to get hard. The lab ones, though, should not be hard because it's all going to be taken from your pre-labs and your post-labs and what you did in lab, what your outcomes were. What is it? Something about the experiment, maybe something about an outcome. It's purely stuff that you physically had contact, touching, feeling, doing. So that will, those sometimes are easier for people and sometimes it's harder because when you look through the lab manual, there's sometimes 11 pages. When you look at the pre-lab business and then the instructions how to do the lab and then what your outcome would mean if you got pink or blue or whatever. So the lab ones will get more difficult as we go on because we have more labs and more detail, more things that the lab is telling us. Yes?
Will you take into consideration for our lab quizzes, like the little tweaks that you've made for our labs, like during section? Like, for example, sometimes we haven't had enough material, or like all of us can't do the experiment exactly the way it's in the lab manual.
Ooh, so you'd have to point out which ones we didn't do exactly.
I don't remember.
Yeah.
It's just like the day of, like,
yeah. The things that I can't take off is, I guess I can. Sometimes we're not brought the same organism that's in the book, but that really shouldn't make any difference. Gram-positive, gram-negative, or gram-positive and gram-negative is all we would need to know. So it's not, I would never ask you, a certain organism was given to you in this experiment, what did that organism do? So that's not the purpose of it. Why do we do this experiment? What would it mean if you got this particular outcome? And it's something that you physically did, and you physically wrote down, and you read in the lab manual. So the only reason it gets harder, because there's so much in one experiment even. There's just a lot of stuff going on. Of course there's a lot of stuff up here, but this is more like little multiple choice kind of thing, and the ones for the lab seem to be a little bit more in depth.
So the quiz is going to be multiple choice?
Well, almost like it was the last time. There'll probably be some pictures of things that'll say what's this, what's that kind of stuff.
And it covers everything after the exam or the first quiz?
The first quiz was only 1 and 2, I think, and then the exam is 1, 2, and 3.
Yeah.
As we had it like almost back to back.
Yeah.
So it would go from 3 to, like, I think we were saying it's 4 and 5. I forgot. Somebody mentioned
Okay.
they thought they
5,
were.
6, 7.
Yeah.
3, 5.
Well, yeah. I'll put it on the quiz thing. And exams will always be a tiny bit different. They will be a little bit more in depth, so that you'll have more explanation that you have to write these. And also because we need to only have a short time to take them. We need every bit of lecture time, otherwise we'll not finish all the lectures. And really they have the lectures planned out so that if you have a certain lecture, that will coincide with the next thing. Because I think the next lab that we have is we have UV that we're doing, so you need to know a little bit about the UV, even though the lab manual will tell you all you need to know to do the experiment. But still, it's nice to have the lecture introduce that to start with. Okay, does that make sense? Okay, so that's all I have. There is a lot of extra slides on here that I thought I had to read it, but I did it really quick at school and maybe I didn't put it on my UV. So, okay, so you want to turn in your little things here? Thank you. So if everybody wasn't here earlier, we did talk about a couple things, but we'll talk about it in lab. Thank you, thank you. Oh, look at a little delivery boy. Yeah.